Thanks very much. It's my pleasure to be here at the JGI user meeting this week. I haven't formally engaged with this community before. There's lots of new faces, which is always good news for DARPA. So we want to sort of get our message out there and get as many people excited about what we do. For those of you that aren't familiar with DARPA, I just want to give you a brief introduction to the agency. So while I'm a scientist by training, I'm a funder by profession. So I look to invest in technologies that should really advance our national security capability. DARPA was started about 6 years ago in response to Sputnik. So that was a complete surprise to the United States. President Eisenhower said that can never happen again when it comes to technology. So let's create an agency that has the resources to both prevent technological surprise, but also create technological surprise. Some of the technologies that you're probably well familiar with that came from DARPA include stealth technologies, Siri, if you use that on your phone, the ARPANET, of course, so the precursor to the internet started at DARPA with just 4 nodes in that network. So where does biology play a role here? 3 years ago, DARPA stood up the Biological Technologies Office as an acknowledgement of the Department of Defense that biology is becoming very critical to not only our economic security, but our national security. So I'm looking to make pivotal early investments to really springboard a field forward to make big changes in types of technologies through programs like Safe Genes that address genomic security, and also living foundries, so having security in our economic and biomanufacturing capabilities. So there will be some metabolic engineering discussion there to whet your appetite with this crowd. So early investment in DARPA, we were one of the first funders of synthetic biology, and we are the largest funder in synthetic biology historically, together with the DOE. We've funded advancements in sequencing and synthesis of DNA. When we started looking at DNA sequencing at the Human Genome Project, that cost about $3 billion to sequence a single human genome. Right now, we're at about $1,000. It takes a couple of days instead of many years, and Illumina and others are in hot pursuit of the $100 genome. Synthesis, we're at the point where we can synthesize entire small genomes, so microbial genomes and viral genomes, but we're on pace to make entire chromosomes and potentially mammalian genomes in the future, in the near future. We can read DNA, we can write DNA, we can now edit DNA with tools like CRISPR-Cas9. I was really excited about 4 years ago when those seminal papers were first published, the types of new opportunities that those tools can bring. But after thinking about it for a couple of days, I also got a little bit concerned about some of the new vulnerabilities that these new technologies bring to us. So opportunities across scales in biology, ranging everywhere from the smallest genomes of viruses to entire ecosystems. These types of technologies, like genome editing, are really outpacing our advancement in biosecurity and biosafety in this area. Just since the invention of CRISPR-Cas9, or the application of CRISPR-Cas9 to genome editing, we've made dead Cas9 with effectors, and then we've already made smaller and better editors like CPF, and there's a slew of new editors, as you know, that are coming out. Derivative technologies, such as gene drives, are also pretty interesting. So these types of tools, you can apply a self-propagating genome editor to biased inheritance. So no longer are you at the stochastic 50-50. You can inherit a trait from mom or dad, but we can bias that with 100% precision to push a trait through. In the case of something like a mosquito vector, what this could imply is an incredible opportunity to push a trait through a vector population that gives us resistance to malaria, or you can push an infertility gene through a population to suppress that population. So for the first time, we have this thrilling possibility of stopping a disease at the insect vector before it becomes a problem for humans. But on the other hand, we now have a tool at our use that could make a population of organisms extinct. This is a very powerful technology that we take very seriously how we move forward with such a technology. The challenges include a lack of controls. Once we start an editor looking at a genome to change that genome, we don't have a great way to turn it off. We don't have any countermeasures. If somebody would try to infiltrate my genome, I don't have a way to stop them from doing that with a genome editor. There's also off-target effects. If we want to pursue aggressively new genomic therapies against cancers or blindness, what if there's off-target effects? If you're taking a drug, you can just stop taking a drug, And eventually that'll leave your system and you can restore to baseline. But if it's actually changing your genome, we need to think more creatively about ways to address those types of challenges. And then finally, I think there's no shortage of gene drive models out there, but there's a real shortage of data to see really how these systems behave over time. And that was really my inspiration in thinking about, we can't really retrofit safety onto these tools, so we need to build them in from the beginning. So the Safe Genes program, the call for proposals went out late last year, was to create new controllers for genome editors, also the first-in-class countermeasures for prophylaxis and really ways to inactivate genome editors in vivo, but tools to remediate genetic changes in environments. We have great ways to clean up oil spills or chemical spills. What about a genetic spill? If there's an engineered gene that is released into environment, we don't have a way with precision to make the smallest change possible to return that gene back to its original state. And that's the pursuit of this program. Unfortunately, we don't have a lot of data, so I'll tell you in the next slides at a high level the types of projects we will be funding, but we'll be announcing it excuse me, this summer. And if you have any ideas, I'm all ears to hear about ways that we can think about using these tools to more safely go out into open environments. From this program, we anticipate the new controls for genome editors, not only for editing the genome, but also the transcriptome and the epigenome. So these will be first-in-class new editors that you haven't seen before. Also, countermeasures that could be small molecules, also nucleic acids and proteins. How can we inactivate editors before they cause problems? And then genetic remediation. We're going to pursue in mammalian systems, we're going to introduce an edit and wait a little bit and then reverse that edit. That would be an incredible capability, I think, to demonstrate if there are off-target effects or nefarious use case, or even accidents. Bioerror is something that I've been concerned about beyond bioterror that we're used to in national security. How can we reverse those changes? And then finally, really generating large population studies to look at the behavior of gene drives over time, over populations. One of my biggest worries is that, not that these things will be wildly successful and cause a species to go extinct, but that they won't work as well as we hope them to. If we think these tools can help us combat malaria and we release them into an environment, what if they don't work, just given the biodiversity of organisms that are out there? We need to be ready and prepared for those types of situations as well. So just to really foot stomp this, these new capabilities will allow us to engineer something like a mosquito genome, release that insect into a wild population and convert the genes and traits in those populations to be resistant to a disease like malaria or Zika. But with safe genes, what we'll also enable is the ability to inhibit infectiveness. If we see some sort of off-target effects in these systems, return them back to baseline and then turn them on and off when we want them and where we want them. That's our ultimate goal. Obviously, with a tool that's so powerful, DARPA is not really in a position to tell the world what we should do with these types of technologies. So we've sought outside perspective to help us address what's the safest way to innovate in this field by getting perspective from experts, from community members and other thought leaders. So we've funded the national academies recently on 2 studies. One is looking at gene drives on the horizon. What's a very safe pathway to move forward? And we've taken the guidance from the national academies and built that into our program. So under Safe Genes, we will not have an open release during the 4 years of that program, but we're going to prepare all the data and get the regulators on board so that if we are successful at the end of 4 years, the community and regulators can make the decision to use this technology or not. And so there'll be continued activity on those topics, and I encourage you all, if this is something that's important to you, to stay involved. You may have also seen the recent National Academies report on human gene editing. So as we develop the tools that we can even edit things like the germline, how should we move forward thinking about those types of edits? This is a very critical question. Some germline human editing change now can impact the human population for hundreds of years to come. So it's kind of a really serious thoughts that we need to put behind if we would ever use these types of tools in those scenarios. So I'm going to shift gears entirely and go into what other types of security that DARPA looks at. As a program manager, I'm looking to make those initial investments to disrupt fields. So oftentimes I go well outside my area of expertise. In this case, in the area of the bioeconomy and biomanufacturing. So the bioeconomy represents about more than $300 billion of our total national economy. We can use microbes, as you know, to make things like fuels and medicines, industrial chemicals, genetically modified organisms to improve crop production. We just heard about that recently. So what's DARPA's role here? So we want to think about, can we take engineering disciplines that DARPA has helped to really innovate and apply some of those principles like design, build, test, and learn, and apply those to biological systems, to make new chemicals that will be of value to us, commodity chemicals, small molecules, specialty chemicals. We know a lot of the metabolic engineering companies are making fragrances, for example. What about material properties? Can we take it even up a level and say, well, rather than a specific molecule, there's something that I want that's really strong or thermally stable. Can we make microbes produce those products for us across many industries? So the approach from DARPA side is the Living Foundries program. We're taking the design, build, test, learn process, breaking it down in the design components to identify a target molecule that you want to make, design a genetic construct. So this would rely, in this case, very heavily on some of the sequences that JGI curates, for example. Build those, so automate genetic construct assembly and insertion into workhorse organisms, for example. And then test those microbes that contain these designs in a very high throughput manner using mass spec and other approaches to see, are you even making that molecule that you're looking for? If you are making it, are you making enough of it? And then learn from that. Sometimes we're going to get those constructs right and be able to make a molecule of interest, and sometimes we're going to get it wrong. But we can learn from those processes so that the next time we go through this cycle, we can do it better and more efficiently. Once we can detect a molecule of interest, we'd like to evaluate its properties and then also scale it up. If we're going to be making molecules that are of value to the Department of Defense and just the bioeconomy at large, we need to make it in most likely kilogram quantities. So I'll walk you through some of the recent innovation and accomplishments of the program, starting with the first phase of the program, which were just the enabling technologies. When this vision first came aboard, we didn't have all the tools that we needed to completely integrate this design, build, test process. So early investment was to build those tools for automation. And then now we're in the current 1,000 molecules component, where we've reached out to the community and just said, what are 1,000 molecules that you really need? And we'll try to make them for you. We're working through that list, and I'll tell you a little bit about that as well. So through the Living Foundries ACG early program, we started with the poster child. This is the amorous example of farnesene, which took 10 years to make at a cost of about $100 million. Some of the nuance to this story is that within the first year of trying to make this microbe at Amyris, they were able to detect farnesene. But it took the balance of that 10 years to really optimize and perfect the flux through that system. Many of you as PhDs probably also have tried to engineer a strain to do something or make something that you want. And this takes many, many years and PhD expertise to look to the literature, mine for the enzymes. How can we do that? So DARPA's question to the community was, how can we do that to not only make tens of molecules, but thousands of molecules on much shorter timelines to really get down to take less than a year to make a molecule of interest at a much, much lower cost. So we invested in miniaturization and automation, lots of robotics to help us do this, also algorithm development that I'll go into in some of the next slides. Developing new engineering tools to do this in a highly multiplexed manner, and then looking at scale up. So this is one of the most critical components and the most challenging. Once you have a detectable quantity, how can you actually make liters and liters of this for downstream testing? So briefly, the punchline for the 1,000 molecules, we're 1 year into a 4-year program, and we've already made 323 molecules at detectable amounts. I don't know if this floors you, but this sort of floors me. We've gone through essentially one design, build, test cycle, and are already that successful. And I'm really excited to see what the program has in the future, and as we move towards scale up, to see what are the new challenges and opportunities we can make there. So how are they making those 323 molecules? Tobias told us a little bit yesterday in his search for carbon fixation enzymes and pathways, you really have to mine through a lot of data to discover, assemble, and insert genes, maybe not only from your target workhorse organism like E. coli or yeast, but really across many different organisms, from human genes to plant genes, and integrate those all together to make molecules of interest. Being DARPA, we spiced it up a little bit and added different types of challenge areas for the performers, looking at either molecules that have already been previously synthesized or are currently extracted at a high cost. This is sort of the lowest bar, so our teams should be able to do that, but let's try to do it better than it's already done today. And then let's look at molecules that are not routinely synthesized, but in theory, looking at the sequence databases, they should be possible given the enzymes that are out there. But then we also wanted to look at molecules that are effectively unattainable. We couldn't either identify enzymes that could synthesize those reactions, or for whatever reason, they're a problem for biology because they're toxic or they're solvents. And the way that we wanted to mine these large amounts of data, some of our performers, Michael Fischbach mostly, has developed tools like Cluster Finder and Anti-Smash that I would imagine this community is well familiar with, to really look at clusters that are in microbial communities to identify gene sets to synthesize some of these reactions that we'd like to achieve as part of this program. And then you can integrate that into the Anti-Smash framework. And this is just an example of what some of those gene clusters would look like, which these could be new candidates of enzymes that we want to use to synthesize some of these reactions. We can put those together and also make predictions on the types of molecules that can be produced. And we can prioritize the different designs and go through those, insert those into organisms, and select the best performers. So our teams have done this. They've made 323 molecules, but they've had those lists from day one, so they've had a lot of time to think about how they would approach putting these tools together. So again, true DARPA style, what we've done is we sort of stirred the pot a little bit, and we did what we're calling a pressure test, where we gave them, at the end of their first phase of the program, a 3-month challenge to say, we're just going to give you the names of 10 molecules. You make them, go. And so I'll just show you the results of one of those projects that MIT took on. We gave them 10 molecules, and MIT asked the question, so not only do we want to make these molecules, but what's the most impactful way that we can pursue these molecules to add value to the communities that are relevant to these molecules? And in one example, this molecule, rebecamycin, is a pretty complex molecule. And what MIT said was, well, we see this as a very valuable anti-tumor agent. There's a lot of commercial applications downstream of this. But we know it's a potent anti-tumor agent, but maybe there's derivatives of this molecule that we can make that are even more potent or more biostable. So when they produced the designs for pathway synthesis, they really focused on these early upstream steps to make a core molecule that could then be derivatized with many different genes that would enable ornamentation of different chemical R groups here to produce 500 unique molecules. So we asked them to make 10 molecules. They came back and told us ways to make 500 new molecules. And I think that it should be obvious that this is of very high value to this team. And MIT is already immediately after the pressure test in collaboration with Novartis pursuing these derivatives for new types of molecules that are not only potential anti-tumor agents, but new antibiotics, new antivirals to pursue. I'd also like to highlight some of the accomplishments of one of our commercial performers, Amyris. So in the middle of this graph, you can see a dendrogram. This is a dendrogram of the about 450 molecules that they're pursuing under the Living Foundries program. The ones in pink are those that they've been able to hit so far within the first year of the program. And the way that they've done it is they've tried to automate as much of the process of the design, build, test, learn cycle as possible, starting with looking at the 469 molecules they intend to pursue in collaboration with RZeta. They've identified about 1,200 reactions that are needed to reach those molecules. So by reaction, that might be a 3-step synthesis pathway or maybe a 10 or 25-step synthesis pathway. Once they've identified those reactions, they select the parts and design the strains. They order them. DNA synthesis, I'll say, is sort of the most challenging part in terms of timelines right now of that MIT 3-month challenge. 2 months of that was waiting for DNA. So if you have new solutions to the synthesis problem, I'm certainly all ears. After ordering those genes, they assemble them. Many DNA assemblies, more than 4,000. They build the strains, they culture them, and then they attempt to measure the product. So they've already measured over 13,000 strains to look for target production. And they're, as you can see, they're pretty successful in doing so. What's most remarkable to me is that they've automated all of the steps of this process except for starting to measure that process. There has to be a human in the loop to optimize the measurements and then for the analysis. Taking that long PhD that might take 3 or 4 years to build a pathway, they can do this within 4 hours. It's really tremendous capability. So what comes next? At DARPA, we're always looking for the next challenge. And what we're running into in the Living Foundries program is, okay, well, of those reactions that you weren't able to make, so they didn't hit every product that we needed them to make, they said, well, it's not effectively attainable because we've run out of uniprot. So this is a quote I take from Patrick Boyle at Ginkgo Bioworks. They just can't find the enzymes that are going to catalyze the reactions that are needed to make those molecules. So I'd really like to challenge this community, and I'm curious to hear your ideas. Can we push into some extreme environments, sequence some new genomes to figure out enzymes that can catalyze new reactions that haven't been catalyzed by, in an integrated way by workhorse organisms for biology to make things that will help sustain life in extreme environments? So as I do work with a military agency, and these are the types of environments that we're either used to living in or we anticipate we will be living in the future. So forward operating bases, there's analog environments, of course, that we can be mining, even microgravity. So there's a lot of interesting work on the ISS. We have a mission to go to Mars eventually. How will we enable life there? I think biology is going to be a critical component of sustaining human life in those environments. And then finally, maritime operations as well. There's many applications for biology in these scenarios. So with that, I thank you for your attention, and I look forward to speaking with you for the rest of the meeting.
All right, thanks so much. Do we have questions for Renee? So how do you initially choose kind of the big domains that DARPA is going to target? Do you kind of convene workshops or something, or is it mostly the people at DARPA who kind of sit down and decide where the priorities are going to be?
Mm-hmm. It always starts with the program manager, but then we sort of go out and talk to the community. We visit universities, talk to people about their research, and then we as a team, we have each program manager has a strong team of PhDs. Actually, one of them's here, Colby. If you're here, raise your hand. You can talk to Colby too about any of these challenges. And we try to figure out what is the investment that a field needs to propel it forward, and then we bring those concepts to our director, and that it's a binary yes/no whether or not we can go forward with the program.
Yeah. Okay, great. Anyone else? Oh yeah, we got someone here.
Thanks. It's a very exciting talk, obviously. I was wondering about the last quote, about the idea that we've run out of uniprot, because in the genomes, all the genomes that we study, there are hundreds of thousands, if you add them all together, millions of unannotated proteins, and many of them enzymes that might have the functions that you're looking for, but the bottleneck really then is assigning function to sequence.
Absolutely. I think that the annotation and knowing what those enzymes do in a better way or high throughput way would be, I think, an investment in this field that could really bring it forward. So I think that's exactly the challenge, right? Thank you.
All right. Looks like we got another back here. Yeah.
Along those lines, I mean, we really know very little about over 4 billion years of evolution of diversity. Mm-hmm. We know about a very small fraction of that diversity. And so is there interest in, joining with JGI and expanding our understanding of biodiversity?
I think there is a lot. I have a lot of personal interest in biodiversity, especially given some of the challenges with the gene drive. I think biodiversity probably in a good way, is going to be one of the barriers to entry of some of these things to be real tools that we could use in the market. And understanding that diversity, there's only a few examples. There was the 1,000 Mosquito Genomes project recently out of the UK, looking at the diversity of just 1,000 mosquitoes that have been caught and collected, and that's such a small subset of what's actually out there. And I think even just understanding the baseline sequence in the environment it would be really helpful if we want to introduce engineered genes into the environment, whether for bioremediation or tools like gene drives. I don't think we have a really great sense of the biodiversity that's out there already. I believe NSF has some initiatives to try to quantitate that.
All right, and we've got
One
someone
more?
back here.
Yep.
Yeah.
Oop.
Yeah, I really like the framework for dealing with the potential negative externalities of genetic modification. I know just some thoughts on that is that techniques that people might develop to undo the genetic modification, it seems like they could also undo natural modification. They themselves could be a problem, and I'm sure you've thought of that.
Mm-hmm. Yep.
I'm sorry, and I forgot the other part, so I'll leave it there.
I agree with you. You could absolutely undo some, for example, inherited diseases. You can undo those genetic errors and replace them with properly functioning genes, or something like microbial resistance that has emerged over time. That would be a great target for reversal. But really, just given the resources that I had available, we sort of made the framework for the project only around engineered genes. But the best DARPA programs are those that really make platform capabilities that we could apply to multiple challenges. So that's a great insight that you had.
All right, well, let's thank Renee for a really thought-provoking talk.